This video covers the material that's in Chapter 1 of your textbook, and most of this material should be review for you. Biology 211 is cell and molecular biology. We're going to cover similar material again this term, but in greater depth. And so this first chapter is just an introduction to a lot of the material we covered in Biology 211. We'll talk about cell properties in this video. What do all cells have in common? I'll talk about the differences between prokaryotic and eukaryotic cells. I'll talk about the endosymbiotic theory or the origin of eukaryotic cells. And then very briefly, I'll make a couple comments about viruses. Cells were first discovered by Robert Hooke and Anton von Leeuwenhoek in the 1600s, but the significance of cells wasn't fully appreciated until the 1800s. Three researchers, Theodor Ch1, Matthias Schleiden, and Rudolf Virchow, were credited with establishing what are called the three tenets of the cell theory in the mid-1800s. The first tenet of the cell theory is that all organisms are composed of one or more cells. The second tenet is that the cell is the structural unit of life. We have not discovered any life forms that are smaller than a cell, and life is built from those units. So organisms, multicellular organisms like ourselves, are composed of many cells that build that larger structure. And then the third tenet of the cell theory is simply that cells can arise only by division from a pre-existing cell. So cells do not arise spontaneously as intact, fully developed cells. On Friday's lecture, I'm going to talk about the origin of cells because cells had to initially evolve from some pre-cellular environment, and we'll talk about thoughts about how cells arose originally. But now that we have established cellular life on the planet, right, we are not getting new cells from arising new. All cells are coming from pre-existing cells. There are several recognized basic properties of cells. For example, cells are complex and organized. They are not just a ball of chemicals. Even the simplest cells are highly organized. We have atoms that are arranged into molecules, molecules that are arranged into polymers, polymers that are going to interact to make more polymers. Different cell types produce different polymers and are consistent in structure and appearance. Bacterial cells have different DNA than human cells, and those cells look different. Human neurons and intestinal epithelial cells have the same DNA, but they make different proteins and they look different. On this slide, the image you're looking at are intestinal epithelial cells, and they are going to look like other intestinal epithelial cells, and they're going to have a predictable structure. They're going to have these structures like these microvilli on the apical surface. Right, they're going to be composed of organized proteins. All those microvilli are going to have similar structures. They're all going to have mitochondria in these particular cells. The mitochondria are on the basal end. So again, here's the apical end of the cell. Here's the nucleus. Here's the basal end. The basal end has the mitochondria. And those mitochondria are also complex and organized structures. They're going to have their own sets of proteins. This is where we produce most of our ATP. Cells also possess a genetic program and the means to use it. Information is stored as DNA. As you know, that DNA is stored in the nucleus of the cell. That DNA must be translated into proteins for cells to function, and that DNA must be copied for information to be passed on to the next generation of cells. Another property of cells is that cells are able to replicate. They are able to divide to produce more of themselves. That's how we go from this single cell, this fertilized egg, to produce a two-celled organism, which will continue to develop to produce this sea urchin embryo or sea urchin larvae. Cells also acquire and use energy. Biological processes are constrained by the laws of chemistry and physics. If you want to build biological molecules and run cellular processes, you need to invest energy. Energy can come from a variety of sources. Generally, light energy is harvested through photosynthesis, and then energy is passed from one chemical to another. But I will briefly talk about exceptions to this during the course. This sea urchin fertilized egg, right, to divide and grow to become a two-celled organism, to become this sea urchin larvae, needs to invest energy to build these structures and to perform its biological processes. Cells also carry out controlled chemical reactions. Harvesting energy from glucose is the same chemical reaction as burning wood. Cells can control the rate of the reaction and collect some of that energy that's released during the breakdown of glucose, and those reactions are controlled by enzymes. We'll talk about metabolism this term. Metabolism is the total of the chemical reactions in a cell. So the breakdown of food sources for cellular energy, the use of cellular energy to build structures in the cell, this is all comprises the cell's metabolism. Cells also engage in mechanical activities. Materials are transported along cytoskeletal elements. Cells move from one place to another. If you look at this sea urchin embryo, these dashed lines in here are cell structures made from cytoskeletal elements. Cells also are going to respond to stimuli. We have receptors that interact with signals from the environment, photoreceptors that detect light, mechanoreceptors that detect physical movement, hormone receptors that detect signal molecules within an organism. Right? Cells respond by changing their metabolic activities, by moving, by dying. All right, there's this complex interaction of cells responding to the external environment as well as the internal environment. Right? As this two-celled organism turns into this larvae, then the cells need to know what DNA to express. They need to respond to their neighbors. Right? This section of the tissue needs to know to become a structural element. This tissue needs to know to become a mouth. This tissue needs to know that it has to become digestive organs. And then this whole organism responds to what's going on in the environment around it. Another property of cells is that cells are able to self-regulate. For example, when a cell is low on energy, it needs to use glucose stores to make more ATP. When ion concentrations move out of an acceptable range, the cells need to adjust the level of ions. When the cell begins to fail, it needs to be able to self-destruct. So cells need to be able to regulate themselves in order to function in a multicellular organism. And the last fundamental property of cells I'm going to talk about is that cells evolve. The origin of cells is a big question of cell biology, but it's largely ignored by textbooks because it is not well understood. We'll spend some time Friday talking about the origin of cells. The textbook I assigned for this class doesn't really address the origin of cells, so I've posted on the course Moodle site a chapter from another textbook that does talk about the origin of cells. And once cells arose, the evolution of cells is fairly well understood. All living organisms evolved from a common ancestral cell. We call this the last universal common ancestor. And all cells have common features. They have a common genetic code. They share a plasma membrane. All cells have ribosomes. All cells have common biochemical processes. We divide cells into two different types based on the complexity of their internal organization. Prokaryotic cells have a simple internal organization, while eukaryotic cells have an endomembrane system and other organelles. Regardless of whether we're talking about prokaryotic and eukaryotic cells, as I just mentioned, all cells share a common ancestor. Evidence of this is that all cells have a common genetic code. They have a DNA sequence, a similar DNA sequence that codes for amino acids. They have common metabolic pathways. All cells have a plasma membrane. All cells use ribosomes to convert their genetic code into proteins. Life on the planet arose approximately 3.4 billion years ago. And so there are different estimates. Your book says that complex prokaryotic fossils arose about 2.7 billion years ago, but estimated that life was around for about 700 million years before those fossils. So depending on which of these bars we're looking at, you could say that life arose about 3.4 billion years ago. There was some precursor to those first cells as we know it, so some estimates say life arose 3.7, 3.8 billion years ago, but at 3.4 to 3.8 billion years ago, life arose on the planet. We have evidence of cyanobacteria around for at least 2.4 billion years. This figure from your book estimates it at somewhere around 2.8, but we know that somewhere between 2.4 and 2.8 billion years ago, cyanobacteria were around on the planet. One of the ways we know this is that cyanobacteria are photosynthetic, and around this time we had a huge increase in atmospheric oxygen. That atmospheric oxygen came from photosynthesis. Eukaryotic cells then arose about 2 billion years ago, and That's when we started to get the more complex eukaryotic cells. We use DNA sequences to figure out how organisms are related to each other. Life has been grouped into three major divisions, or what we call domains. And when you look at the relationship between organisms based on their DNA sequences, we have some surprising findings. For example, what we now know is that animals and fungi are more closely related to each other than they are to plants. So that means you and yeast are more similar to each other than yeast are to corn or you are to corn. Similarly, another surprising finding is that the archaea are more closely related to eukaryotic cells than the archaea are to bacteria cells. Archaea and eukarya share a more recent common ancestor than the archaea and bacteria do. However, despite the fact that archaea and eukarya are more closely related to each other, we group the bacteria and archaea together into the prokaryotic cells. So these bacteria and archaea are the prokaryotic cells, and we group them together because of their cellular structure. They are less complex cellularly than the eukaryotic cells are. Those prokaryotic cells have a simple internal organization. They're usually described as not having the internal structures that are found in eukaryotic cells. For example, eukaryotic cells have a membrane surrounding the DNA. The DNA is held in a nucleus. Prokaryotes do not have a nucleus. Eukaryotic cells have membrane-bound organelles that compartmentalize functions. Eukaryotic cells do not have these organelles. Right? Prokaryotic cells have no nucleus. The DNA is in the nucleoid. There's no boundary membrane. There's no organelles in prokaryotic cells. The DNA is as a single circular chromosome. And they often have other small rings of DNA that they pick up from other bacteria, which we call plasmids. But their chromosome is this one circle of DNA. Eukaryotic cells, in contrast to those prokaryotic cells, have a complex endomembrane system. And they have the Golgi complex as part of the endomembrane system. These vesicles are part of the endomembrane system. We have a smooth and rough endoplasmic reticulum that are part of the endomembrane system. These eukaryotic cells also have mitochondria, and if they are plant cells, they have chloroplasts. Eukaryotic cells also have a very extensive cytoskeleton that's used for motility, contractility, support. They have multiple linear chromosomes, and they have a very complex cell division process called mitosis. Table 1.1 in your textbook outlines all the differences between the broad differences between prokaryotic and eukaryotic cells, and you should be familiar with those properties that are found in Table 1.1. Eukaryotic cells originated from prokaryotic cells, and I'm going to start talking about the endosymbiotic theory, the theory of how eukaryotic cells arose. And if you really understand that endosymbiotic theory, the question of which types of cells are more biochemically diverse, prokaryotic cells or eukaryotic cells, is fairly intuitive. So, prokaryotic cells have many different cellular mechanisms for using energy. Eukaryotic cells, however, are more limited. Eukaryotic cells are either organotrophs. Organotrophs get energy by eating organic matter, or phototrophs. Phototrophs use energy from light to make organic molecules. So all eukaryotic cells are either organotrophs or phototrophs. Prokaryotic cells can be organotrophs, they can be phototrophs, but many prokaryotic cells are also lithotrophs. Lithotrophs use organic molecules to get the energy to make biological compounds. So they can get their energy from hydrogen sulfide, they can get their energy from hydrogen, from iron compounds, from elemental sulfur. And so prokaryotic cells as a group have many more different sources of energy from the environment. They can get environmental energy in several different pathways, whereas eukaryotic cells are either limited to being organotrophs or phototrophs. Additionally, plants can only fix carbon dioxide, whereas there are prokaryotic cells that can fix carbon from carbon dioxide and other prokaryotic cells that can fix nitrogen. So there's a lot more biochemical diversity in prokaryotic cells. Now, how does this relate to the origin of eukaryotic cells? Well, all eukaryotic cells are derived from one prokaryotic cell that ate another prokaryotic cell, but then, rather than digesting that prokaryotic cell, formed a symbiotic relationship. So here we have one anaerobic prokaryotic cell. This cell could perform glycolysis. It can convert glucose to pyruvate and use that reaction to generate ATP. But glycolysis, breaking down glucose to pyruvate, is not particularly efficient. You don't get that much energy out of that process compared to aerobic respiration, where you take that glucose and you break it down through the process of oxidative phosphorylation and generate a lot more ATP. So this anaerobic, inefficient bacteria consumed an aerobic bacteria or aerobic prokaryote that was undergoing oxidative phosphorylation, and instead of digesting that bacteria, they form a symbiotic relationship, and those aerobic prokaryotes are now what we know as our mitochondria. And so the reason eukaryotic cells are less biochemically diverse than prokaryotic cells is because when this occurred, there were dozens of different prokaryotic cells, all of which had that had all sorts of different types of metabolic processes, different ways of acquiring energy from the environment. But eukaryotic cells are derived from a small subset of that original population. All eukaryotic cells are derived from a prokaryote that was anaerobic and has one type of aerobic bacteria that is now a mitochondria, and all those other different types of energy metabolic processes that exist in the prokaryotes are not represented in eukaryotic cell line. So after several millions and millions of years, that original eukaryotic cell created lots of descendants. Some of those descendants became our protists, our fungal cells, our animal cells. However, one branch of that line underwent another round of endosymbiosis where a photosynthetic bacterium was engulfed and ended up becoming another organelle, the chloroplast. So plant cells and algal cells are the result of a second round of endosymbiosis where they picked up chloroplasts. What is the evidence for this? How do we know that this is how mitochondria and chloroplasts arose? Well, first, mitochondria and chloroplasts have their own genome. And the genome of mitochondria and chloroplasts is small, with circular chromosomes, with a genome similar to that of bacterial cells. Mitochondria and chloroplasts also have their own ribosomes. Right? All cells have ribosomes to make proteins from DNA. Mitochondria and chloroplasts have their own ribosomes, and they all have a double membrane. The outer membrane is from the host cell that engulfed the free-living bacteria, and then the inner membrane is from that free-living bacteria that got engulfed by the host cell. Our prokaryotes and mitochondria are both about the same size. I'm going to, in the next slide, give you some estimates of how big different cell structures are. I want to mention now that you should be familiar with these terms for different sizes. So you should be familiar with the abbreviations and the relative size for meters, centimeters, millimeters, nanometers, angstroms, as well as picometers. You would expect if mitochondria were formerly free-living bacteria, they would be about the same size as prokaryotic cells. Mitochondria are 2 micrometers in diameter. Prokaryotes are typically 1 to 5 micrometers in diameter. The nucleus of eukaryotic cells is about 5 to 10 micrometers in diameter, and eukaryotic cells are typically 10 to 30 micrometers in diameter. However, the largest can be up to 10 meters long. So you can have neurons in some large mammals like whales and giraffes that go from the spinal cord the whole way out to the periphery, and you can have neurons that are up to 10 meters long. However, most of our cells are restricted in diameter to roughly 30 micrometers for eukaryotic cells. You can have some cell bodies up to 100 micrometers or even 500 micrometers. But still, that's relatively small. Why do cells tend to be so small? Why are we made up of trillions of small cells instead of being made up of a handful of large cells? There's 3 primary reasons for this. The first is that the bigger the cell, the longer it takes to produce cell structures. If you think of the nucleus as a factory for producing mRNA, it's much quicker to produce the necessary materials if you have 100 small cells with 100 nuclei that are producing mRNA than it would be for one big cell with one nucleus to produce all of that necessary mRNA. Another problem is that the surface area to volume ratio decreases as cell size increases. Cells need to take in food, they need to remove waste. Big cells have a smaller relative area for exchange. They have less surface area relative to their volume in order to exchange waste and bring in nutrients. And then the rate of diffusion is inversely proportional to distance squared. So what that means is that as distance increases, it takes proportionally longer for things to move out of the cell. So the bigger the cell is, right, as the cell gets bigger and bigger and bigger, it takes longer and longer and longer to diffuse nutrients in or waste products out, and so it takes much longer for exchange to occur. I'm going to end with a couple final points. One point I want to talk about is that most of the experiments we're going to talk about this term are going to be performed on model organisms. We use model organisms because cells are similar, so when we learn something about one type of cell, that can tell us about other cells. But also, model organisms tend to be organisms that are easy to maintain in the lab. It's easy to reproduce them, to grow them. And Escherichia coli is a model bacterial cell, a prokaryotic cell that is small, easy to culture, and easy to manipulate. Saccharomyces cerevisiae is a model organism for studying eukaryotic cells. This is a yeast cell. It's a single cell with a relatively simple genome. And the interactions between regulatory genes, even in a simple eukaryotic cell, are complicated, but we can use this simple yeast cell to start to understand those processes in other eukaryotic cells. Arabidopsis thaliana is a model for understanding plants. It's small. It can be grown easily indoors. It produces thousands of offspring per plant. It only takes 8 to 10 weeks to reach sexual maturity and has a relatively small genome for a plant. Cenorhabditis elegans, C. elegans, has a short life cycle. It only takes several days to reproduce. It's small, it's easy to breed, has a small genome for an animal. That's a nematode worm. Drosophila melanogaster is an arthropod, a fruit fly. It only takes 9 days to reach sexual maturity. It's small, it's easy to breed, has a small genome for an animal. And Mus musculus, or a mouse, has a very short life cycle, is small, it's easy to breed, and this is a model organism for understanding mammalian cell processes. You should know these names, you should recognize these organisms because they're going to be coming up again and again as we're talking about cellular experiments and what types of organisms these experiments were performed in. And then the last couple points I want to make are about viruses. So viruses are acellular, they are not cells, but they have a big impact on cells, and they are used for a lot of cell biology. So viruses are small, relatively simple, obligatory intracellular parasites. They are unable to reproduce, metabolize, or perform biochemical processes independently of a cellular host. They do contain their own genetic material, and they have a protein capsule, what we call the capsid, around the outside of the virus, but that genetic material codes for making new viruses, but the virus can't make new viruses without using a host cell to do all those biochemical processes for it. Viruses have a protein on the surface that typically binds to a host cell surface. They enter host and they cause the host to make viral proteins and more virus material. Once again, when we pick up on Friday, what I'll be talking about is the origin of cells. So where did the original cells come from? And we'll start talking about the processes that are necessary to make life and where those processes might have begun.